I also had some doubts whether I should focus on a particular disease model. I decided to show several of the neurodevelopmental disease models that we are studying. And the reason I decided to do it is because I think we can learn a lot by comparing disease models that share some symptoms. There are a lot of issues on comorbidities in neurodevelopmental disorders, and I think that looking at different disorders from different perspectives, maybe we can get better insights. Of course, there is a trade-off here. This means that I have a little bit too much data to show, so I will try to be light. So if you have questions about the details, you will have to ask me afterwards. So I will start. Just to say that most of the results I will show you have been produced here in Portugal at the headquarters of the National Brain Imaging Network, where we have several facilities like a research-dedicated MR, 3 Tesla MR scanner. We also have molecular imaging facilities like a cyclotron and PET, which enable us to study from a multimodal point of view neurodevelopmental disorders, which I will focus today. So being here, I will talk you about a disease model called neurofibromatosis type 1, which we have studied from the neuroanatomical, from the sensory processing point of view, and also from the point of view of neurotransmitter regulation and their impact on cognitive function. And this model has actually been inspired by the work of Rui Costa, which is one of the PIs here at the Champalimaud Foundation. We have studied this model from the point of view of sensory processing, and I will tell you in a minute why. I don't have time to detail the different visual functions that we have addressed in this model. So this model, as I said, has been inspired by the work of Rui and Alcino Silva many years ago, and basically what they postulated is that there is excessive RAS signaling, which leads to excessive inhibitory neurotransmission. And they have shown that such excessive GABAergic inhibition leads to impaired spatial memory and working memory. Since inhibition is very important for visual function and sensory function, we started looking also at visual function in this model. This is also very interesting because knowing the mechanism that leads to impaired inhibitory neurotransmission, you can think about therapeutical approaches, which I will not have time to address today. So as I said, we use several techniques, so functional MRI, spectroscopy, and anatomical methods to elucidate this phenotype. And one of the things that we noticed in our functional imaging study is this network that is hyperactivated when you have stimuli that activate a lot the visual periphery, which we call the magnocellular pathway. This pathway has actually been shown today by Antonio Damasio and seems to be overactive even for very simple visual stimuli in this condition, so the default mode network. And we were quite intrigued by this finding. In fact, you see that this network, which is related to introspective activity, mind wandering, it's not just that it fails to deactivate, it actually overactivates during the task performance. And so we were quite interested in this. So it looks like these patients are mind wandering when they are doing a task. And this is, of course, not good. And this might be related to attention deficits that these patients also have. We know that one quarter of these patients have attention hyperactivity disorder. So not all have this clinical diagnosis, but during this study we find a lot of evidence for attention deficits, so like as in a continuum. And so our hypothesis, which is actually related to hypotheses in other neuropsychiatric disorders, that this network fails to deactivate during task performance, seems to also hold true for this condition. We also studied this disease from the neurophysiological point of view. So you have this very simple eyes closed, eyes open paradigm to study the alpha rhythms. You know that when we close the eyes, there is a 10 hertz rhythm that increases in the posterior part of the brain and means that the visual brain is in a needle state. And interestingly enough, we notice that these patients have too much alpha. Even when they open the eyes, the alpha should go away, but it remains. So there is a failure to eliminate this alpha rhythm when they open the eyes. And again, very recently, it's becoming more and more clear that excessive levels of alpha are also related to attention. So we used a very complicated paradigm. Visual paradigm. I will not explain it in detail. But basically, we manipulated contrast, both color contrast and chromatic contrast. We studied visual responses, and we also study how alpha modulates during visual stimulation and task performance. I will not maybe detail too much. We found a deficit, a change in contrast responses. But most interestingly, we found, you see this small peak here, around 10 hertz. We found that even during visual stimulation, there is too much of alpha rhythm. So alpha rhythm is suppressed during visual stimulation, but they have a lot of alpha even when they are doing a task and are being visually stimulated. So this seems to be like a trait that is very, very, very constant in this condition. And you might ask, what are the behavioral implications of this finding? To make the story short, because there is not much time, we replicated in this group something that is already known in controls, that too much alpha is related to misses. You miss something in the visual world. You have to do a detection task, and if there is too much alpha, you miss your target. That's basically what these plots show here, both for controls and for patients. When you have more alpha, you have a higher incidence of misses. You miss a visual target. So how does this relate to the GABA hypothesis? So far, we have functional imaging and neurophysiological evidence for changes in the brain that might be related to attentional deficits. But how does this relate to the GABA hypothesis? Because this is still the open question. So to answer this question, we developed for the Siemens scanner we have in Coimbra a technique to measure this small peak, this GABA level, the GABA concentrations in the brain. We measured it first in the visual cortex, and we were surprised to see that actually the GABA levels are reduced in the clinical population. Let me say that what we are measuring is a measure of the presynaptic pool of GABA. And interestingly enough, there is a genetic link to this reduction of GABA levels. So it's mainly the mutations in the neurofibromin gene that are related to missense mutations or splice site mutations that lead to the lower GABA levels. And this is a significant genetic effect. This means that if the organism is producing a truncated protein, it will interfere with the normal protein and lead to lower GABA levels. So there is really, we can really have a genetic to neurochemical link. And this would also explain the variability in the cognitive phenotype that these patients have. And in fact, in this disease, it has been quite difficult to make solid genotype-phenotype correlations. But in fact, this was a quite clear one that we could make. Okay, you might ask, well, you are measuring static levels of GABA. Does this really relate to a functional phenotype? Interestingly, these GABA levels are inversely correlated with brain activity. So this is really a physiological marker. It predicts the level of visually evoked brain activity. So this GABA level is really something that relates to a functional marker. We also looked at brain oscillations. I already told you about one type of brain oscillation, the alpha, the one that is related to distractive behavior, too much alpha, too much misses, too much distraction. But we also looked at other brain rhythms, the gamma. I'm sorry that I don't have time to explain these plots. These are just spectral plots. Here you have the frequency, here you have time. These oscillations that are between 40 and 80 hertz are what we call the gamma oscillations that are related to cognitive function. And we found an interesting correlation between the frequency of these cognitive oscillations and GABA, but that is only present in the controls. It was already known from the group of Cardiff that our neurochemical repertoire, mainly our GABA levels, can predict the frequency of these rhythms, of these oscillations. But interestingly enough, these correlations are lost in the patient group, meaning that the neurotransmitter levels are no longer controlling the frequency of these oscillations. And these oscillations are quite important because they have been claimed to be disrupted, these cognitively related oscillations, to be disrupted in conditions such as autism and schizophrenia. But we know that neurofibromatosis type 1 is also related to learning problems, so we wanted to look at more high-level functions. And we developed, I have to say that this is mainly the work of a PhD student that finished, Inês Violante, and also Maria Ribeiro, a postdoctoral fellow in our lab. They developed a go, no-go task, a very simple one. You have to refrain to press a button when there is a 3, but you have to press a button when any other number appears. And so basically, this is actually a paradigm where you can, a paradigm for studying impulsivity or motor impulsivity. And basically, we found that the patients have a lot of errors, both of omission and commission. What does this mean? Omission is that when you should press a button and you fail to press the button. Commission is that when you should not press the button and you have pressed impulsively the button. And what we have found is that there is a correlation. We found, of course, a correlation, an inverse correlation between reaction time and errors, proving that this is really a good paradigm for impulsivity. And then we measured GABA levels in the brain, but now in this region of the brain, in the anterior cingulate. We found that GABA is also reduced in this part of the brain. We also did neurophysiology and found that these potentials, these P2 potentials, both in the GO trials and in the no-GO trials, are reduced in the patient. So there is a neurophysiological marker. And in fact, to make sure that this marker is really related to correct responses or not, when responses are correct, the green curve, the potential is higher. This means that the reduction of these potentials in the patient's group may really mean that they are related to this behavioral phenotype. And interestingly enough, we found a correlation between the levels of neurotransmitter and this potential. There is an inverse correlation between GABA levels and the amplitude of these potentials. So here we can really say that we find a relation between behavior, functional markers, and neurochemistry. So there we really find a relation between neurochemistry, a physiological marker, and behavior. I will maybe skip this part. We are also doing research on algorithms for medical diagnosis. If we can use this data to separate between patients and controls, these are just data mining algorithms. So since we have a lot of imaging and neurophysiological data, we could potentially use them as a diagnostic algorithm markers, but I don't have time to show this. This is published in Human Brain Mapping. We can use structural data both in children and adults to differentiate between clinical groups, but then the question remains. So if I have reduced GABA levels presynaptically, and if the hypothesis is that there is excessive inhibition, how can we explain this? Our hypothesis is that this is a developmental disorder. Professor Damasio talked about homeostasis. So maybe there is regulation happening during development. So our hypothesis is that you have excessive postsynaptic inhibitory activity, and to compensate homeostatically for this, you have less available GABA presynaptically. So we are now measuring using PET. So we are basically using a GABA-R receptor marker, quantitative PET. We are quantifying GABA receptors in the brain. This is ongoing work, so to be cautious, I will not tell you the results. It's quite sure that it's not reduced. We are still trying to see if it's increased or not. But to give you an example of our prediction, I just show you another molecular imaging measure. This is of D2 receptors, raclopride, yes, D2 receptors. And you see this is, you basically measure the striatum. This is in control, this is in Parkinson. And look here at this Parkinson patient. You see that there is a lot of postsynaptic receptor in the caudal part of the putamen. This is exactly where you have presynaptic loss. So our prediction is that if you have a lot less presynaptic activity, you have postsynaptic hyperactivity. And that's what we expect to find in neurofibromatosis type 1, but I cannot yet give you the answer to this question. So now, to make an interim summary for this disease, we have found neurophysiological and neurochemical markers that correlate to cognitive dysfunction in this group. We still need some more research to elucidate the nature of the neurochemical phenotype, although we can already see that there is a genetic link with this phenotype. We are, actually this disease has been related to a more broad spectrum of neurodevelopmental disorders, mainly because of the attention deficits. Now we found, as I showed you, evidence that there might also be an impulsivity phenotype, but we are mainly interested in social cognition disorders. So we have been doing research recently in Williams syndrome and in autism disorders, and we are developing actually paradigms based on attention to social stimuli. You know, if somebody is looking at a given direction, if one of these avatars is looking at a given direction, a normal person will follow the attention of that avatar. And that's what we call a social attention cue. And we are developing neurophysiological paradigms, actually P300 types of paradigms, to measure these social attention cues, to guide, to actually also for neurorehabilitation purposes, to try to guide using brain-computer interfaces the attention of the person that is looking at these displays. But to be able to do this, you need to classify each event. You know, each time the avatar is looking somewhere, you need that the person identifies this. And for this, you need statistical classification tools that anytime there is something changing in the brain, in the neural signal, that is related to the detection of something happening with the avatar. And for that, we are doing statistical classification of single events. I don't have time to explain. Just to say that with very few trials, you can know, just based on the brain signal, if the person is following the attention of the avatar. We have built a lab in the pediatric hospital of Coimbra, a virtual reality lab. In fact, we have a model of the room. The person can put glasses that are and feel, they can walk in the room, so the real world and the virtual world are absolutely identical. And we are doing that exactly to train these social attention skills. In the same lab, we also have eye tracking techniques. I just show you these are actually used in the conventional neuropsychological assessment. And we are doing motor tracking, neurophysiological measures, and eye movement measures. You see here, these are just heat maps of where the people are paying attention to. And you see that they are not paying attention to, they are paying attention to objects, but not to the people. That's very typical in autistic spectrum disorder. So we are really generating a phenotyping database that will be very important to characterize subphenotypes in autistic spectrum disorders. This is kind of one of the setups we have. So there is actually, the person can interact with an avatar and learn social attention skills using this technology. But now I go a bit more simple. If you look at this display, you don't see anything. But if I make it, if I make it move, I hope you see a mask. You see a head, right? So this is what we call coherence. You see the head because all the dots are moving coherently, right? This is what we call a structure from motion paradigm. You perceive the object because there is motion. And this very well illustrates the concept of, a very simple concept of coherence. You see something because something is coherent. And of course, we are using these paradigms because we are very interested in the central coherence hypothesis in autism. If I show you the next movie, you probably will not see much because we have degraded the mask. So we use this type of stimuli in Williams children, a very rare but very, very inspiring model in cognitive neuroscience. It's also genetically determined, but we all have characterized all these patients. And this is a very interesting study that we published in PLOS ONE to illustrate the concept of central coherence. So if you have here an hierarchical figure, a P made of A's, and you have here a triangle made of arrows. And this is the drawing made by a Williams syndrome subject. You see that he can see the A's, he can see the P's, but he cannot integrate both. So you have a local global deficit here. With the triangle is even worse. And interestingly enough, these stimuli hierarchical Navan figures have been used very often to characterize central coherence phenotypes in autism. And then we have the hypothesis, if this is true, then the autistic phenotype should be the one to show the strongest deficits. But in fact, we found the strongest phenotype in Williams syndrome, and these are the 5 groups we have studied. We studied around 100 kids. So this is autistic spectrum disorder with intellectual disability, autistic spectrum disorder, no intellectual disability, chronologically age-matched groups, IQ control groups. And you see that in the autistic spectrum disorder groups, they are relatively preserved in this central coherence task. And so we think the link between central coherence and the autistic hypothesis might have to be revised, or at least we would expect then at least concerning this marker, we would expect that the more autistic group which would have the strongest central coherence phenotype. So I will skip this because it's too much. Then we have, you have seen these masks, these objects moving, these faces. We have done neurophysiological studies to see how they differ between controls and patients. You see that there is a huge neurophysiological difference. So the patients, we tried to make the behavioral task matched. So we made it simple enough that the patients could do it, but they can perform normally, but the neural response is very different, and the neural generators of these responses are very different. So what does this mean? That this suggested to us that the brain is being reorganized since they need a different network to perform this task. So these are just the principal component analysis. We see that there are very different components in the patients compared to the controls. But then we wanted to look at spectral signatures. Sorry again for this complicated slide, but I will try. It's simple. You have frequency here, you have time here. And if you compare controls, and Williams syndrome looks very different, right? You could tear them apart by just looking. So Williams syndromes have a lot of activity at 40 hertz. The controls have a lot of activity at 60 hertz. They could not be more different. So we really find totally different pattern of brain activity. And what does this mean? So this is what is highlighted here, very different patterns of activity. So this means that they are probably using a different part of the brain to solve this task. They are totally reorganizing their brain. And to understand that, then we did an fMRI study to see how the brain responds to these objects that you have to see motion to perceive the object. And to make the story short, because I don't have much time, you see that the Williams syndrome activates a lot of the medial part of the brain, what you call medial dorsal stream. And actually, the control group tends to activate more, you see here in the difference map, the lateral part of the brain. So it looks like we use more the dorsolateral part of the brain to integrate these surfaces. And the patients, since this part is no longer working, they reorganize to the medial part of the brain. And this was actually quite striking. You see it again here. This part of the dorsal stream is not working in the patients, and it's only activated in the control group. So there is really reorganization that allows to keep performance preserved. which is, I think, an important message concerning neurodevelopmental disorders. This is made clear again here. Patients activating more medial part of the brain and not activating this lateral part of the brain. So there is really brain reorganization. These I skip. This is again the same thing. You see, these are controls. These are, and you see that the pattern that you see in the groups are quite different. So this is Williams syndrome. These are controls. Just your eyes sees that is different. But then the question, here we are measuring amplitude of the responses. But what about each part of the brain talking with each other, what we call brain coordination, or what was told before, functional connectivity? How do different parts of the brain talk with each other? This is what we call synchrony. And if you look at synchrony, then the controls have a lot of synchrony, you see, not the patients. And you see that exactly for where you have less activity in the controls, that's where you have more synchrony. So we were completely missing a feature of the brain. You know, people tend just to look at the amplitude of responses, and sometimes they fail to think about how different parts of the brain talk with each other. And so in this model, we found a loss of synchrony that you actually see here. These are synchrony lines in controls. The patients lose a lot of the synchrony. And so this could be another mechanism that could explain the loss of integration, the loss of coherence in this disease model. I will not have, of course, you would ask, well, I told you that there is an oscillation at 40 hertz that is different in the patients, and an oscillation at 60 hertz. But this seems a bit abstract. You would like to know where they come from. So we did an EEG fMRI study. Unfortunately, I can summarize it very briefly. And we found out that the 40 hertz oscillations come from the anterior insula that are related to perceptual decision-making. And the 60 hertz oscillations are mainly coming from the visual cortex. So different oscillations correspond to different cognitive functions, and therefore should be interpreted differently. In fact, we think that the patients, because they have difficulty, more difficulty in doing the task, they activate more the anterior insula and recruit more these 40 hertz oscillations, but with less synchrony. This is just, I will not detail it. Can you see faces here? Maybe here, some of you can see faces. Here it's an inverted faces, here is a guitar, here is scramble. We use this stimuli to try to understand where these oscillations come from. They come from different parts of the brain and subserve different functions. And this helps us explain what is the problem with our patients. So I think I have to finish. This is just the brain maps with our simultaneous EEG fMRI studies. You see here the visual cortex, and you see here the insula for the 40 hertz oscillations. For the 60 hertz oscillations, you see mainly the visual cortex. This was proven doing simultaneous EEG fMRI. I'm sorry if I'm not acknowledging everyone. This work involved many people, well, mainly Inês Violante and Maria. I don't know where she is, for the neurofibromatosis study. For the Williams syndrome and autism, Susana Moga and Inês Bernardino. And of course, the people at the pediatrics hospital. And for the neurochemistry, Richard Adams from Johns Hopkins, and the Cardiff University for the genetic studies. Thank you for your attention.